Hi, everyone. I'm Trent Blizzard, the president of the North American Mycological Association, and I am here tonight with Julie Daoust. Say hi to us, Julie.
Hello, everyone. Excited to be here.
Yeah, and we're going to get a nice presentation from her here in the upcoming hour on functional mushroom supplementation as a prebiotic fiber impacting microbiome composition. So there, I nailed the title for you. Um,
You did. You did.
So Dr. Daoust received her Ph.D. in natural product chemistry from the University of British Columbia and has worked in the nutraceutical industry with a focus on plant-based products. Her research has been centered on bioprospecting for new functional metabolites in plants, fungi, and bacteria, and on clinical outcomes related to plant-based nutrition. Dr. Daoust is the Chief Commercial Officer at M2 Ingredients Incorporated. So I think without further ado, I'm going to turn it over to you and turn my video off. And if you see me come back and wave, and it's probably because we have a question. So take it away, Julie. Thank you.
Yeah, thank you, Trent. I'm excited to be here today to cover some of the incredible work that the team at M2 Ingredient has done over the last couple of years related to digestive health. Here is our two topics that I'm very passionate about. One, functional mushrooms, and two, the microbiome and its impact on our health. And M2 Ingredient is a company that was founded by Steve Farrar, who's a mycologist that's here on the call today, and Sandra Carter. We're based in Vista, California, and we grow 10 species of organic functional mushrooms. And I'm very privileged to work with a team of brilliant mycologists, chemists, food scientists, and food safety and quality specialists. It's been quite a ride to be part of the functional mushroom industry here in the US. Lots happening, lots of interest, and more importantly for me, a lot of exciting research. I grew up foraging for mushrooms with my family, and we also prepared some of our own preparation using botanicals as I was growing up, and we started finding lion's mane and reishi in the forests in Canada where I lived, and it got me really interested in functional mushroom, the traditional usage of these species. And I continued using them personally in my life, in my diet. And when I started my PhD in natural product chemistry, we were looking for new molecules that could have interesting effects. And mushrooms are partly the next frontier in terms of drug discovery. There's a ton of different pharmaceuticals today on the market that were derived from fungi, but there's more to discover. When we think about mushrooms, mushrooms are essentially huge bioreactors. They're very fragile physically. When we think of the mycelium, for example, it's one cell layer across with no physical defense at all. And so the organism essentially wages a chemical warfare on its environment to protect itself from bacteria, other fungi, parasites, or to be overgrown. And so leveraging that chemistry and the bioactive molecules that these organisms produce is really fascinating and really the next frontier in discovery for not only pharma, but functional ingredients and other biomaterials. So really excited that there's a budding interest here in North America on studying these organisms. I think we need more mycologists here in North America. So I know I'm preaching to the choir here, all people interested in mushrooms, but if you can encourage all young people around you to study mycology, you know, there are professional opportunities out there for mycologists, and we need more people studying these incredible organisms. Today, I'll cover a little bit of information related to the traditional use of some of these species we'll cover today for digestive health. I'll do a quick overview of some of the recent research that relates some of the species we'll cover with impact on the microbiome, and then I'll dive into the research that we completed with our research partners at M2. Would love to answer questions. I'll try my best to answer them. These are big topics, big fields, so hopefully I can answer some of them. And if not, that might give us some food for thought for future research. So lots of interest currently in using functional mushrooms for all kinds of functional benefits, right? We talked about the fact that mushrooms are essentially bioreactor producing all kinds of bioactive molecules with all kinds of benefits. Mushrooms are closely related, and fungi in general are closely related to animals. And so we share a lot of the same cellular pathways as fungi. We breathe in oxygen, we exhale CO2, we're heterotroph, meaning we need to eat carbons in food. We can't fix it from the atmosphere like plants do. And because of all these similarities, we actually do share a lot of cell receptors with mushrooms, which makes them, again, such a great source of potential functional benefits. And so you see here, consumers are seeking all kinds of benefits from mushroom, and that's because there's a lot of diversity in mushrooms and the functional benefits that they'll provide. So digestive health is one of the top benefits that people use mushrooms for today. And some of it is anchored in traditional usage. So lion's mane was traditionally used in Chinese medicine, mostly for its digestive health benefit. So it wasn't necessarily tied to cognitive health like we think of lion's mane today. So today, maybe not, the bulk of the research likely focuses on cognitive health, but originally it was really used as a tonic for digestive health. Cordyceps militaris was also used to support digestive health, to regulate kidney function, and as a tonic. Reishi, the mushroom of immortality, one of the most widely used mushrooms in traditional Chinese medicine, was used also to support many things, but digestive health was part of the usage case for reishi. Mushrooms, we know are considered a prebiotic ingredient at large. So when you consume mushroom, when you eat them, you'll consume a lot of different types of fiber. So some soluble, insoluble fiber, beta glucans, alpha glucan, chitins, and other carbohydrates. So including mushroom in your diet is a great way to include more fiber, and they are prebiotic ingredients. So a lot of people don't necessarily like to eat mushroom. I encourage everyone to include lots of mushroom in your diet because as Americans, we don't consume enough fiber. So what better way to include fiber in your diet than through mushrooms? Mushrooms as prebiotics are thought to have their effect as a prebiotic tied to a few different molecules. This isn't an exhaustive list, but chitin and beta glucans have a prebiotic benefit in mushrooms. So beta glucans and chitin are found in all cell wall of fungi. So they're found in all fungal part. They're an integral part of the fungal cell wall. Beta glucans will vary in some of their branching and structure. So not all beta glucans are made equal. They're really a class of polysaccharide, and the table on the right just highlights some of that diversity of structure. For a long time, we thought that chitin couldn't be digested by human, but recent research showed that chitin can actually be digested in our gut. So we produce an enzyme called chitinase, that the gene for chitinase, for the production of chitinase, are often turned on in individuals that are exposed to chitin. So either through consuming mushroom or insects, or even some types of some shellfish. And so by habituating the gut with a little bit of chitin from some of these sources, chitinase will be produced and that can be digested. When we cook mushroom or heat them to prepare some of these functional mushroom preparation, we'll usually break up the chitin to some degree, and then the chitinase that we produce in our gut can help break down the chitin. And chitin has been shown to be a prebiotic fiber, so it increases the diversity in the gut microbiome, but it doesn't seem to stimulate the short-chain fatty acid production. And we'll talk about why that's so important in a moment. Chitin has many benefits as a prebiotic fiber. It's immunomodulating, and it can also, has been shown to increase insulin sensitivity and therefore have an impact on metabolism. So an important molecule or class of molecule that's found in fungal tissue, and something that until recently we thought we could not digest. On the flip side, beta-glucans are a type of fiber that's digested by our microbiome, and they're considered a prebiotic fiber. They affect the diversity of the gut microbiome, but they also promote the production of short-chain fatty acid. They're also considered an immunomodulating molecule. So let's cover a little bit of recent research that's looked at the impact of some functional mushroom on the microbiome. So I'll present a number of different studies. Some were in animals, some are clinical trials. So this first one was done by a group that did a mice study, and they wanted to understand the impact of reishi on sleep in mice, as far as whether or not it was a microbiome dependent effect. So what they did, and what we see in these graphs, is they looked at the sleep incidence, the sleep latency, the sleeping time, so markers of sleep quality, and they look at 5-HT concentration. And what we saw is that the bars in green here are the reishi treatment. So reishi did indeed improve the markers for sleep quality in these mice. But when the reishi was consumed alongside an antibiotic that would affect the microbiome, right, the composition and the total biomass of the microbiome, that effect was not significant. So that's the purple bars we're seeing in these graphs. And that's leading us to believe that the impact of reishi on sleep may very well be microbiome dependent. In this other study, here we're covering a couple studies. So one was an animal study on mice, looking at lion's mane and the impact of lion's mane in the microbiome of these mice. And what was seen there is that the lion's mane would increase a number of beneficial bacteria that help produce short-chain fatty acid, and therefore were related to reduction of inflammation and potential improvement in cognitive health. The lion's mane also helped decrease some bacteria that were not beneficial and that would have impacts that are negative to cognitive health. A clinical trial in 2004 also looked at a number of different outcomes related to lion's mane consumption, but what they noted is from the lion's mane consumption, they saw an increase in the diversity of microbes in the gut for the participant. This was in human, and an increase in short-chain fatty acid production. And they associated or positively correlated those two outcomes with the production of neuropeptide Y and brain-derived neurofactors levels. So really interesting study that really piqued our interest into the impact of some of these species on the microbiome. And the last one I wanted to cover was a study that also looked at mice using cordyceps extract and cordycepin. And essentially in this study, it's a bit of an eye chart here, so I'll summarize best that I can, and hopefully it makes sense to all of you. So these mice were pushed into a diabetic state through high-fat diet. And the chart on the left here, the colors indicate diversity of microbial species in the gut. And so the more color you see, the more diverse the population in the gut was. And what they saw is that at both high and low dose, the cordycepin samples did improve the biodiversity of the microbiome in these diseased mice. Same with the cordyceps extract, at a similar effect. And then they looked at different outcomes of metabolism, and they saw a decrease in oxidative stress, cholesterol, and liver enzymes for both treatments, the cordycepin and the cordyceps extract. So all these results really encouraged us to understand what would be the impact of the strains we grow at M2 Ingredients and understand their mechanism of action. So following some of these publications, we decided to embark into our own research. So I'll just cover a little bit what our product is, because there's a ton of different products on the market that are relevant for all kinds of usage. So I'll just cover a little bit what we do. So we grow all of our species on something called solid state fermentation. So we grow on oats, and our species are grown in a bioreactor bag. And at the bottom right here, you'll see this is a picture of our cordyceps. So this is the growing block, and essentially we'll have the mycelium colonize the grain and then form those beautiful cordyceps fruiting body. And this product would then be harvested and milled. So we don't produce extracts or products or whole food mushrooms. And so all the research that you'll see today was done on these products. And we're really focused on understanding the efficacy of these species and furthering the research so that we can continue to invest and understand how they can help with their functional benefits. So on the market, you'll see a lot of different products. Some are fruit body only, some are grown on liquid fermentation. So there's a product on the market called CS4. It's a Cordyceps sinensis product that's been on the market for a really long time. And it's popular in a lot of sport formula. So that's grown in liquid fermentation. A lot of the turkey tail polysaccharide products that are used a lot in research and in Asia are grown in liquid fermentation. So we grow on solid fermentation, and the work that we're presenting here today, those species were both mycelium and fruit body of the various species. So the work I'm presenting today, it was done in an artificial gut model. And that's typically how you'll study microbiome impact, especially for screening like we did. And that's also how you can define whether or not your ingredient is a prebiotic. So we wanted to understand, are our mushrooms prebiotic? Do they have those effects? Do they fit the definition? And at the dose that we recommend on our products and for our customers. So we partnered with a group called ProDigest. They're a world specialist in microbiome research and in helping different organizations understand whether or not an ingredient's a prebiotic. So across the world with the different nutrition organization, there's not a lot of complete alignment on the definition of a prebiotic, but at large, a prebiotic would have to do some of the impact that I'll list here. And a lot of people confuse prebiotic and probiotic, so I'll just take a minute to explain that. So the prebiotic is the food for the probiotic. So prebiotics is a fiber essentially that the microbiome, so those organisms in your microbiome, would consume. When you buy a probiotic on the market, so a bacteria, some are found widely in your gut already, so you're essentially increasing that population by taking the probiotics, and others are created in the lab or found in nature and found to have probiotic effects. So here today we're talking about prebiotics, so that's food for those microbes in our gut. So when we did this experiment, essentially it's an artificial gut model, but we implant the microbiome from real people. So we collected, well, we didn't personally collected it, thank God, but the fine folks at ProDigest, who chose an odd career, collected samples from healthy individuals. So we had a half male, half female population. They consumed a Western diet and had not taken antibiotics in the last 6 months. So meaning that their microbiome, we could assume, was stable. So these folks didn't have also any kind of gut conditions such as IBS or Crohn's disease. So they were healthy and consumed a balanced diet. So what makes a prebiotic? Well, a couple things. First, we need to see a lowering of the pH in the colon in the artificial gut model. And that indicates that there's lactate being produced, which indicates that there's the beneficial bifidobacteria in the microbiome, which is good news and beneficial. All prebiotics, all fiber will produce gas when consumed by the probiotics, so the microbiome. And so that's why we get bloating or gas produced when consuming beans and high fiber foods. Some fibers are much harder to digest than others. Inulin is considered a gold standard as a prebiotic. It's used in all kinds of formulas, but a lot of people have a hard time digesting it because it produces so much gas in the gut. So you want that to show that it's a prebiotic, but you don't want too much of it. Then you want the material to increase the production of short chain fatty acid. And short chain fatty acid have all kinds of beneficial impact on our health, not just in the gut, but otherwise. And we'll talk about butyrate specifically. Butyrate is very important for gut lining health. So keeping that gut lining nice and healthy so that it doesn't become permeable and allow the entry into our bloodstream of all kinds of nefarious molecules. We also see that butyrate has an impact on cognitive health and the blood-brain barrier. So again, very important. It has impact on immunity and inflammation, so meaning it has impact on recovery as well. So from a sport performance perspective, it also has a role. So in the nutrition community, butyrate is a big topic and has been for the last couple of years. So there's a ton of research currently that's focused on butyrate, much outside of what we care about here in the functional mushroom world, but it's an area of accelerating discovery. Then you want your prebiotic to increase your overall microbiota, so the beneficial bacteria in your gut. And typically they'll have a pretty selective impact on the community composition. So meaning a prebiotic will enrich the microbiota of certain species. And that's reasonable because different microbes will prefer certain foods and then establish themselves. And you want that to a degree because pathogenic bacteria can more readily overcome a very diverse population, whereas if you have very well-established population of beneficial bacteria, typically it'll be harder for those pathogenic bacteria to overtake your gut. So that can make you more resilient. And then we also looked in this study at the impact of some of these mushrooms on the metabolome. So our gut microbes produce a ton of different molecules, including serotonin and GABA and dopamine. So a lot of our feelings molecules, our neurotransmitter, are actually produced by the bacteria in our gut, which I didn't understand until a couple of years ago. I mean, talk about the gut feeling being real, right? These microbiomes are necessary for our health, not just for the functioning of our digestive tract, but also for our mood and our cognition. So super interesting work. So we tested a number of our different mushrooms along with also a blend of our mushroom. We call it Myco Multi. It's a blend of the 10 species we grow here at M2 Ingredients. And we saw that indeed these ingredients increase the total beneficial bacteria biomass in the model, meaning they met one of the criteria to be a prebiotic. Then we looked at the impact of these ingredients on the production of short-chain fatty acids. And we saw that indeed we did increase the short-chain fatty acid production and all 3 short-chain fatty acid were increased in the test, which was really exciting because typically you'll see impact of some of these ingredients on one or another, but not all 3. So we were very pleased with these results, and it encouraged us to do some further research into their impact. So then we looked at, okay, so what different species are enriched with each unique mushroom? And what we saw was that each mushroom really had a unique signature. And this speaks to the fact that it's not just a fiber story or a beta-glucan story. There seems to be other compounds in these organisms that give them that unique fingerprint to support the growth of specific population in the gut. what was really exciting was to see Cordyceps militaris supported the growth of some species of Eubacterium and Roseburia, which are important butyrate producing bacteria. And so they're beneficial bacteria that you do really want in your gut. And we were surprised to see that Cordyceps militaris really had such a great impact on those species. And it also had the highest butyrate response in the model. So we definitely wanted to dive further into this. And the results for micro-multi or blend of 10 species is an example of why you need to keep humble in science. The team really wanted to test the blends, and I didn't want to because I thought, well, it's just less of each of the species we grow, because we were all products were tested at a 2-gram dose. So I thought, well, there's just less of each of the species, so we'll have a muted effect. We're kind of wasting money by testing this. And thankfully, I have a great team that are very open-minded and remind me to keep humble every day. And what we saw was that actually this blend had a very broad impact and a strong impact on enriching positive species, beneficial species in the gut. So we decided to dive deeper into its mechanism of action and understand its benefits. So we looked at the impact of cordyceps and this 10 species blend on the gut lining. And there's a model called these CACO2 cells. And what you do essentially is you measure resistance across the cell wall to understand the integrity of that cell wall. So this test is done to understand if compounds or different foods may cause leaky gut, right? That inflammation response on the colon cells that kind of force them to split apart, and that then allow different molecules to cross into the blood that shouldn't cross into the blood. And what we saw was that cordyceps and this micro-multi had a significant benefit on the health of that lining, that gut lining. We also looked at specific markers of inflammation that are associated with leaky gut or the health of the colon lining. And what we saw is that some of our mushrooms, specifically cordyceps, but also the micro-multi to some degree, actually reduce pro-inflammatory molecule and also decrease some anti-inflammatory cytokines. So very exciting results. And then we looked at, well, let's compare these two species of mushroom, or these two mushroom products with inulin, which is again the gold standard in fiber supplements. So you find inulin in a lot of different products. Right now there's a bit of a craze of prebiotic sodas like Poppies and Olipop's. And I can't remember which of the two, but one of them uses inulin, and then you see inulin in a ton of meal replacement formulas and supplements of all kinds. And inulin does increase butyrate production in the gut, so we wanted to compare those mushrooms compared to inulin. And what we saw was that over the 12 days of the experiment, the mushroom increased butyrate production over time and continuously, whereas inulin seemed to plateau and produce less butyrate. And again, it's so important to have butyrate be produced in the gut because it really protects that gut lining and has impact on memory, focus, and mood even. We also saw that inulin produced lots of gas, but our cordyceps and our micro-multi produce significantly less gas in that model, meaning that it would lead to less discomfort than inulin would. Some people can digest inulin quite fine. I'm totally perfectly fine with it, but a lot of people report having digestive upset. So we wanted to understand that as well in this assay. This is again a bit of an eye chart, but essentially what we did is we looked at both the cordyceps and the micro-multi blend at day 5 and at day 12 against inulin to understand what effect it had comparative to inulin. And we saw again that cordyceps at day 12, so at the end of the supplementation, had significantly increased the population of Eubacterium and Roseburia, those great butyrate producing bacteria. And that effect was not only significant to the control, but also to the positive control, which was inulin. So very exciting. And the other part that was great to see was that cordyceps also helped decrease the population of Pseudomonas A, which is a pathogen that can be found in the gut. So not only did it increase the population of beneficial bacteria, but it helped reduce population of pathogenic bacteria, which is a hallmark of a good prebiotic. And this was, again, against the negative control and inulin. We talked at the top of the presentation about lion's mane being used traditionally for digestive health and not cognitive health. When we embarked in this study, we wanted to understand, well, how does lion's mane impact the metabolome in the gut? So those neuroactive metabolites, either neurotransmitter or precursors for neurotransmitter that are produced in the gut. So this is, again, a bit of an eye chart, but what it shows is that when fed lion's mane, the microbiome from these participants produce higher level of dopamine and GABA, which are associated with mood and cognitive health. And it also increased the level of tryptamine in the metabolome. So speaking to the cognitive effects of lion's mane may be a multi-pronged effect, meaning we know a lot about those diterpenes, the arenacenes and erythronones, erythrins, and so on that have an impact on BDNF and nerve growth factor, right? Which is a totally different mechanism of action than what we see here, but potentially lion's mane may have an impact on that microbiome to then promote cognitive health through the gut-brain axis. And these results point to that. So that was really exciting findings, and we're diving more into that. So with that, I went through it very fast here, but hopefully we have lots of questions and can have a engaging discussion in the time we have left together.
Thank you, Julie. Hi, thank you.
My pleasure.
I don't know whether we'll have many questions here or not. Sometimes we get a slew of them, and sometimes we get crickets.
What is late for those East Coasters?
I know it is. It's hard to find a time that works
For
for
Yeah.
everyone.
Mhm.
And I fear we've been training all of our webinar people this year that they can just go watch the video later. Okay, so Michelle popped a question in there. Can we use mushrooms as a prebiotic when making yogurt instead of inulin?
Yeah, absolutely. I mean, that's a good idea. Mushrooms, like we talked about at the top of the presentation, have all kinds of fiber benefits. Here we presented data on our own mushrooms, at the strains we use and at the dosage we recommended, but you can definitely use mushrooms that you consume in your diet or if you make powders of the mushrooms you collect or that you grow, absolutely they'd be a great prebiotic to include in yogurt.
Keith asks if we do a double extractions at home in our own lab, are there any good ways to calculate or consider dosages, at least to a thereabouts?
Yeah, that's a good question and kind of a somewhat complicated one. So the products we tested here are whole mushroom preparation that are dried. We tested them at a 2-gram dose and saw these effects, so the equivalent of a 2-gram dose. I would say that you definitely want your water fraction in this product because that's where you'll get some of those polysaccharides that are going to extract. unfortunately, you're going to lose a lot of them in the mark, right? So, depending on what mushroom you're using, you might want to add the mark or part of the mark back in because you do want that blend of both soluble and insoluble fiber, I think, to get these effects. Um, so yes, a dual extraction could work. I would add the mark back in. And as far as equivalency and dosage, it would kind of depends on the mushroom and you know what it is. But hopefully this gives you a lead of somewhere to start from.
Joshua asks, is there a benefit to mushroom products that are fresh versus, I guess, dry?
Fried, I don't think they're fired, right?
Yeah, I'm going to say dried was what he meant to say.
Yeah. Um, yeah, absolutely. I mean, the fiber will be there and the benefits will be there, whether it's fresh or dry to some degree, as long as it's dry at mild temperature. I mean, I'm guessing a big part of the effects we saw is through the fiber and the polysaccharide fraction of the mushroom, but there seemed to be more to it, right? We saw that each mushroom had a unique fingerprint. So because of that, I think if you process your mushroom at mild temperature, it would be best. As far as fresh mushrooms, some mushrooms are kind of tricky to consume fresh, right? We do need to heat them a little bit to burn off some molecules that may be harmful to health. So it really depends on the species in terms of whether or not you consume them fresh. I always tend to cook my mushrooms or at least do a dehydration step with a little bit of heat to keep things safe. And then Kelly is saying, I'm curious if you compared butyrate production comparing your mushroom and a butyrate supplement such as biobutyrate. We did not compare to butyrate supplements. We were really looking at whether or not we could promote the microbiome itself to produce that butyrate. And again, we have a whole food mushroom approach to this and really wanted to use that whole foods to create that functional benefit. Let's see, did I miss anyone? Doesn't look like it. All right.
I have a question or two for you, Julie. One
Great.
is just a big kind of general question. Like, where does mushrooms as a nutritious food kind of end, and where does medicine begin?
Oh boy. I think part of our way of thinking and realm is driven a little bit by regulatory in a sense. You know, in the US, we think about supplements versus food, and it's really blurred as far as, you know, where does a supplements start and a food end, right? King Trumpet has all kinds of benefits for health, but it's a delicious food and it's sold in the grocery store. Same with lion's mane. Lion's mane is a great example, right? You find it in the grocery store, you found it in the woods, you eat it as a food, but you also see it sold in the supplement aisle. So I think nowadays we really think of things maybe a little bit much through a regulatory framework, and obviously, you know, at work and so on, I need to think this way. But really, when I think of my diet, I really blend things that came from a traditional usage with, you know, more modern ways of growing them and blend them with different foods. So, and I mean, all foods have a big impact on your health, right? We need to eat more fiber, so we need lots of fruits, vegetables, and mushrooms and whole grain. So, you know, you are what you eat, and your food is so central to your health that the line is blurred is the answer.
That was a tough question, I know. Hey, we have
Yeah.
a few technical questions flowing in here. Let me read one to you. Um, Aiko asks, in the slide showing the presence of beneficial bacterium, can you explain why the combo would not produce a bacteria that is produced by 3 out of 4 of the individual mushroom species? Example, eubacteria. Is it just a quantity proportion thing?
Um, Sorry, I missed a bit the question because my Great Pyrenees dog is barking very loudly outside the door. So
Can you
hopefully,
see it in the Q&A from
um,
Iko?
Michael, no, weird. I see Chris Bailey's question and Max Reinhardt's.
Oh, that's the chat. Can you open the Q&A?
Oh, okay.
I can put it
Um,
in the chat for you too.
yeah, please.
Okay, I dropped it in the chat.
Right. So that speaks a little bit to why I was so surprised, right? At the effect of the blend of the 10 species. It seemed to have a signature that was really unique. I think it's no problem about the long question. I think it's probably dare I say maybe there's a bit of a symbiotic relationship between the different mushroom that's creating this effect. Obviously, the eubacterium with cordyceps was at the equivalent 2 gram dose. So maybe that higher dose really promoted those bacteria more, whereas the blend of the different mushroom provided a polysaccharide mix that was preferred by more species overall. So that's how I can explain it. Obviously, we need to dig deeper to understand like what exactly in the product is causing those specific issues.
Do
And
you
then,
do you want to take a look at the chat? I see several
mhm.
questions rolling in there. Even before I posted mine in there, you might want to pick the ones out that you want to answer.
Yeah, so Chris Bailey is asking if I think that the promotion of gut microbiota by fungi will have any specific benefits on disease of the gut, such as IBS. So I do believe so. So the uh, gut lining model that we looked at definitely showed an improvement in the cell health on the gut lining that would essentially prevent the split of the colonocyte to allow inflammation to enter the bloodstream. So it could very well have an impact on IBS. We haven't studied disease state, right? The cell health was done in a model. And following this research, we're actually doing a clinical trial, which is underway, and we're looking at digestive health outcomes in healthy individual. Because we sell supplement and food ingredients, we can't actually, well, we could study disease population, but we couldn't address it in the product that we offer. So we're really focusing on healthy individual and hopefully preventing inflammatory issues. Are we not getting as much prebiotic if we use liquid tinctures as opposed to dried mushrooms? Well, I mean, there's definitely going to be a difference there because when you're doing a liquid tincture, you're taking away some of the insoluble fiber that's in the product, and that typically has some effect on the benefits, although it's more of a bulk effect, so meaning like moving bulk through the digestive tract than a true prebiotic effect, but there's still some things there. So I would say there's still benefits of using liquid extract, but definitely you need to have water in that extract in this particular to get a prebiotic benefit because polysaccharides are at the center of some of that benefit. And then is there also a benefit to using the entire mycelium versus just fruiting body product? I mean, that's a big question. The kingdom of fungi is very broad and very diverse, so I don't think we can make broad generalization that could get us in trouble, right? As a scientist, we always try to avoid doing these things. What I'll say there is that the mycelium is a bioreactor. It is the most fragile part of the mushroom, and it produces a lot of different bioactive molecules that in some cases are not in the fruit body and in some cases are. Again, it's a broad kingdom. So I would say there's some of those polysaccharides in both fungal part, so definitely both could be beneficial. I can only speak to the work that we've done, and that was done on a blend of both the mycelium and the fruiting body, and that's the benefits we saw. But there's definitely benefits of all fungal parts. Again, it just depends on the species and what you're trying to do with it as well, right? So if you're trying to do a product for cognitive health with lion's mane, based on what we just saw in terms of the prebiotic benefit, but also what we know about the aranacines and the ericerins and the ericinone, having a product that includes all the bioactives that the mushroom can produce could be beneficial, because then you're having a multi-pronged effect on cognitive health through the gut and also through the diterpenes that are the nootropic compounds. Let's see. I think I addressed all questions.
Did you get the dairy question? Did I miss that?
Oh no, I didn't. Does dairy have a negative impact? Well, it depends, I guess, on the on the human and what else is going on in the diet. We didn't test with dairy. We will have some information about impact on the microbiome from these mushrooms in the clinical trial that we're doing right now, and there'll be questionnaires about the diet that people in the trial had. So maybe we'll get some clues there, but we haven't looked at dairy specifically.
Okay, well, there we go. Thank you. Much appreciate you spending an hour with us this evening, and sorry about the dog noise in the background.
Oh, I'm sorry about the dog noise.
Yeah,
My dog was barking just outside the door here.
we
So
both have dogs here that are hungry
yeah,
or anxious or something.
very worried about our safety. Yeah. Well,
Okay,
thank you for
well,
tuning
so
in.
thank
Appreciate the
you.
time.
Okay, good evening. Good night, everybody. I don't know how to end the screen yet here.